to describe the three types of muscles. And as you should know from anatomy, we divide the muscles in three types. Okay, skeletal muscles, cardiac muscles, and smooth muscles. So yesterday we described a little bit of the anatomical and morphological features of the three muscles, and from a morphological point of view, we have seen immediately the differences, the anatomical and morphological differences between the three muscles. Okay, from the first one, the skeletal muscle, that is well organized. Okay, in striation, you can see here the striation. They are maintained in the cardiac muscle, but they are lost completely in the smooth muscles. Okay, but also the structure of the two muscles, or the three muscles, are different. Okay, for example, we just said that the skeletal muscle are long fibers, long muscle fibers. Okay, you can appreciate it here, a long muscle fiber, or here. This is just one cell. It is the muscle fiber. Here, for example, in the smooth muscle, you can see the lack of striation, but also the fibers, the single cell muscle fibers that are smaller, okay, compared to the skeletal muscle. So yesterday we start to see differences, anatomical and morphological differences between the three muscles. Also, the localization of the muscle is completely different. Okay, the smooth muscle, for example, we have seen that it's present in the internal organs, in tubes like the gastrointestinal tubes. Okay, or vessels. They have completely different localization and control. The skeletal muscle is under the voluntary control in response to the somatic motor neurons, starting from the cortex, the cerebral cortex, and then the cardiac and smooth muscles are under the autonomic innervation control, so the autonomic nervous system. And the control is involuntary. Okay, so let's start to talk about the first one, the skeletal muscle, because we are almost ready to understand how the contraction takes place, and also how a motor neuron from the cortex can activate the muscle. Okay, now the skeletal muscle represents about the 40% of the total body weight, and as we said before, the function is to move the skeleton. Okay, from a pure physiological point of view, as you probably remember from human anatomy, the skeletal muscle are attached to bones by tendons. Okay, tendons, and it's possible to define the origin of the tendon. Okay. And the insertion of the tendon. The origin is the closest part, closest to the trunk, or to more stationary bone, like this one. Okay, in the insertion here, the more distal or more mobile attachment. Okay, so they have a different type of localization. In this condition, an important point that we have to consider after the definition of origin insertion is the condition of flexor. Okay, the flexor brings the bones together, while the extensor moves the bones away. And this is there are two different muscles, okay, paired from or antagonistic muscle group. Okay, you cannot contract at the same time the flexor and the extensor, obviously. Okay, and for this reason they are called antagonistic muscles. Okay, flexor and extensor pairs form antagonistic muscle group. When one muscle is contracted, the biceps, the other biceps is relaxed, and we are doing. Okay, let's see when you are engaging, you are doing some exercise. Okay, let's analyze again the next slide. Okay, because the antagonistic muscle define a special kind of physiological regulation. Antagonistic. We have found in physiology an antagonistic control of something. Do you remember some antagonistic control or something that we have already studied? We have talked about it. Antagonistic. What is antagonistic in physiology? A part of muscles. Another control that we have seen. Negative feedback could be something like antagonistic, but antagonistic muscle groups are two different, for the muscle, two different movements, and they are, when one is working, the other is not. So, what have you seen up to now in physiology, in all the things that we have learned until now? Here's another thing that is antagonistic. When one is working, the other is not. For example, the insulin and glucagon. Remember, the insulin and glucagon. When insulin is working, the glucagon is not. So it's an antagonistic control. Obviously, we are talking about muscles here. So antagonistic muscle groups moves bones in the opposite direction. Muscle contraction can pull on a bone, but cannot push a bone away, obviously. When you are doing exercise, for example, with some weight, okay, the biceps muscle contract, okay, and is the flexor, so you can lift a weight, okay. But when the extension muscle is working, like the triceps here, the muscle contract, and you have to contract the muscle in the opposite way. So this is the typical antagonistic muscle control. Who is the neuronal part that control this movement? It's the cortex, because this is a voluntary control. So always when we are doing this kind of exercise, or just moving a chair in the table, we contract muscles because we want to contract muscles, okay? Because it's a voluntary control. And in the next video, in the next slide, yes, we will see a video about all the things that we are going to learn in this chapter. Okay, this part, the yellow part here, you already know everything about the movement of the action potential from the hillock to the terminal of the axle. And you know perfectly how the movement is the action potential can be regulated point by point or in a saltatory way. Okay, so now we have to see the neuromuscular junction in a deep way. Okay, so before entering in detail on the structure and function of the neuromuscular junction, let's try to understand in general what is a muscle and how a muscle can respond to electricity, okay? Because this is an electrical stimulation of the muscle. Again, a typical question during the exam can be neuromuscular junction. Can you tell me something about the neuromuscular junction? And again, you can talk about the neuronal part and the muscle. So you have plenty of things to tell me. But let's see what is a muscle. Okay, what is a muscle and how a muscle is constructed. Because the muscle, the skeletal muscle, responds to an electrical stimuli only because it's constructed in a particular way. As I told you before, and probably you already know the structure and the anatomy of a skeletal muscle, but a brief reminder is always helpful. The skeletal muscles are composed of muscle fibers. These are the muscle fibers. And from a physiologist, okay, we are absolutely without fantasy in our mind. So the muscle fiber is a muscle cell. Okay, muscle cell is muscle fiber. It's the same. It's like the cell that we draw always like a circle. Okay, it's a muscle fiber. And the amount of intracellular ions and extracellular ions are more or less the same. Okay. The important aspect that is different is that the amount, the quality, and the quantity of proteins inside of the muscle cell or muscle fiber is different. So, as is depicted here in this slide, we have different names for all the components of the cell. The muscle cell is called muscle fiber, as I told you before. The cell membrane is the sarcoplasm, and the modified endoplasmic reticulum is the sarcoplasmatic reticulum. You perfectly know what is the function of the sarcoplasmic reticulum, right? What is the function of the sarcoplasmatic reticulum? To work as a store for calcium ions. And the amount of calcium ions inside of the sarcoplasmatic reticulum is like 1.5 millimolar. Okay, so the sarcoplasmic reticulum in the skeletal muscle is extremely important because it's a store of calcium ions. And as you remember, for contraction, calcium is extremely important. The muscle can be contracted only when calcium is present inside of the cell. So there should be an important regulation of calcium ions inside of the muscle fiber. The muscle cell, muscle fiber, okay, are long, you can see here, long, cylindrical, and in parallel disposition. A lot of muscle fibers, muscle cells, all together, they create a muscle fascicle, a bundle of fibers. You can see here, a lot of muscle fiber, a lot of muscle cells, all together forms a bundle of fibers. And why? Here you can see a lot of fused, a lot of nuclei. Why? In the muscle fiber, you can appreciate here, a lot of nuclei. Because the muscle fiber are fused cells with many nuclei created by the fusion of many individual embryonic muscle cells. And you probably analyze it in anatomy. This is called syncytium. Because in embryonic phase, all the muscles fuse together. All the muscle cells fuse together. The nuclei are not fused one to the other one, so the muscle fiber is created by the fusion of many individual embryonic cells, muscle cells, and the nuclei are visible here. Remember, the smooth muscle or the cardiac muscle that we have seen before contains a muscle fiber, is built by muscle fiber with one nuclei. Okay, here the situation is completely different. Have you analyzed this point in human anatomy last year or no? Or this is just the first time?
It was this week,
In the day. Okay. So, again, as you can see here, here, muscle fibers are bundled together into fascicles surrounded by connective tissues. This is a connective tissue all around the muscle bundles. Okay. The fascicles here, okay, contains also collagen, contain also elastic fibers, contain nerves, contain blood vessels, obviously, because you can see here and here. It's obvious because the muscle needs to be continuously perfused with blood and also with the nerves that are going to do a neuromuscular junction with the muscle. So muscle fascicles are composed of individual muscle fibers, and numerous muscle fascicles compose a skeletal muscle. And you can see here, you see muscle fiber all together forms bundles of fibers, and all together build a skeletal muscle. This connective tissue surrounds the entire muscle, okay, continues with the connective tissue sheets, all the muscle to the bone with the tendons, okay? There is also another type of cell here, okay, that is called satellite cells. Satellite cells that are something like the stem cells of the muscle. Stem cells of the muscle, they are called satellite cells. Obviously, they are present when we are young, okay? In older people, they disappear, obviously, okay? And their function is to differentiate into muscle for growth or repair the muscle when it is necessary. But as I told you, by the age they tend to disappear. So we described in general the structure of the skeletal muscle. Can you stop me out there? Yeah, and again, this is the resume of the skeletal muscle that describes all the points that we already analyzed. But there are more structures in the skeletal muscle that we have to analyze in order to understand how the muscle can be contracted. So this part, okay, is more the anatomical point of view of a skeletal muscle. In fact, we already know this portion of the skeletal muscle from human anatomy last year. But now we have to deeply analyze this part, that is the cellular physiology of the skeletal muscle. We have to see what is contained inside of the cell, inside of a muscle fiber. We will already recognize that the muscle fiber, muscle cell, okay, are composed by other types of different structure. We already discussed about the different name recognizable here in the red box, okay? But muscle fiber, muscle cells, have the special name that we introduced before. So, sarcolemma for the cell membrane, sarcoplasma for the cytoplasm, multiple nuclei, okay, and the sarcoplasmatic reticulum. Sarcoplasmatic reticulum, that is the endoplasmatic reticulum in other cells, okay? Then we can see also the T-tubules here already, longitudinal tubes with enlarged ends that are called cisternae, okay? We will see soon a figure, okay? T-tubules that are functionally linked to the sarcoplasmatic reticulum. And we already know that the function of the sarcoplasmatic reticulum, such as we can say the same for the cisternae, the function is to concentrate and sequester the calcium ions. Instead, the new cellular part of the muscle that we have to describe right now is in the green box. Okay, this is the new real important point of a skeletal muscle. Now we just define the structure, and then we will see them in the figure, in the next slide. Okay, so first of all, there are the myofibrils. This one, myofibrils. Myofibrils is the main intracellular structure in the striated muscle. Myofibrils. As we said before, the myofibrils is a highly organized bundles structure of contractile and elastic proteins that carry out the work of contraction. The myofibrils, here in pink color, salmon color, is the main intracellular structure in striated muscle. And we find it only in skeletal muscle and cardiac muscle. It's a highly organized, as I told you, highly organized bundle structures of contractile and elastic proteins that, at the end, carry out the work of contraction. And as you already noticed from the figure, okay, the myofibrils are formed by numerous proteins, okay? And you can see here the troponin, the actin, the tropomyosin, the myosin, the myelin, the titin. Okay, these proteins are the structures, the contractile and elastic proteins that at the end carry out the work of contraction. The actin forms the thin filaments, and the myosin forms the thick filaments. So this is the protein part of the myofibrils that is organized, highly organized inside of the cell, of the muscular cell, that are able to generate the contraction and then the tension. Okay, a force is able to do any type of work. Okay, contraction. Obviously, there are also other parts of structures inside of the myofibrils, not only proteins. Obviously, there are ions, glycogen granules, or mitochondria, are extremely important for the production of ATP. Okay, and the energy that you already know from the biochemistry classes. So in the green box, there are the proteins able to contract the muscle. I would like to spend a few more words for the sarcolemma and the T-tubules. This is an important part of the muscle fibers. okay, extremely important structure that is named transverse tubules. T is because it's called transverse tubules, okay, T tubules. The T tubules are membranes. Caprini. Prego, ci vediamo. They are membranes, okay? The tubules are membranes because they are sarcolemma that moves from the outside of the cell, okay, to the inside of the cell. So they are membranes in continuity with the sarcolemma that goes inside and deep in the muscle. Obviously, when we contract a muscle, we have to contract the entire muscle, not only the external part of the muscle. It's quite obvious. And we will see that these T tubules are functionally linked to the sarcoplasmic reticulum, where the calcium is stored. So the T tubules are in continuity with the sarcolemma, and two T tubules plus two flanking terminals form the cisternia. We will see the cisternia plus T tubules are called together a triad. We will see right now. And their function, the function of the sarcolemma and the triad, is to allow the action potential to penetrate nearer the internal structure of the fiber. For this reason, the T tubules goes so deeply in the muscle. Let's see one figure. Okay, this one. This is the structure of the skeletal muscle fiber, where we can recognize all the structure that we just named. Okay, the T tubules in yellow here. The T tubules are component of the sarcolemma that goes inside of the muscle, from the outside to the deeply inside of the skeletal muscle. We can appreciate the sarcoplasmic reticulum, again, where the calcium is stored, and the sarcoplasmatic reticulum with the T tubules, okay, here and here, form this structure that is called triad. The triad is constructed by sarcoplasmatic reticulum cisternia, the T tubule, and the other part of sarcoplasmatic calcium store. So this is a triad. This is a triad. We can see also the thick filament, the myosin filaments, and the thin filaments, the actin. And as you probably know, the contraction of a skeletal muscle is due to these two filaments, thick and thin filaments. It's not like an elastic. Okay, it's just filaments that move one into the other. Okay, we will see. The T tubules here, you can appreciate here the triad. Okay, T tubules and two terminal cisternia, here and here. The triad is made by one cisternia, the other cisternia, and the T tubules. And the T tubules are extension of the cell membrane, the sarcolemma, that associate with the end, the terminal cisternia of the sarcoplasmic reticulum, where the calcium is stored. And you can see also here the thick filaments and the thin filaments. Okay, so you can understand immediately what is the role of the T tubule and how this role is diffusion. Okay, let's see this page. No. T tubule. Okay, as I told you before, the membrane potential, that is determined here outside of the muscle, needs to be transferred inside of the muscle, deeply inside of the muscle, if we want to contract the muscle. So the role of the T tubules is already obvious here. The T tubules are structures that bring the action potential into interior of the muscle fiber. There should be something here that is able to move an action potential from the external part to the internal part of the muscle. And how is possible to do that? How is possible that an electrical signal move from the outside to the external part of the muscle, deeply inside? We didn't analyze this process yet, but probably you should say something about how it's possible to transfer an action potential from here to here. We didn't study yet this process, but how is possible for you to move an action potential from here to here? What is necessary to move an action potential from here to here?
Maybe like you can consider the external part as like the post-synaptic neuron, and so like the action potential will be regenerated.
Exactly, perfect. This is just an external part of a cell, a muscular cell, but this is an external part. So as she said now, there should be something like in the action potential that is able to move from here to here. What is this something now?
Receptors.
Receptors, or what is the important component that moves an electrical stimuli from one part to the other part? An electrical component that moves current. Is the same component that we have seen in the neuron. What is
voltage?
Exactly something voltage? Okay. Until now, the answer is something voltage. Is okay. The example is different, but now we just have to understand that it's something voltage, something able to feel that a depolarization occurred. Okay, great. So these T tubules are able to bring this action potential from the external part of the muscle to the internal part of the muscle. Okay. So this is the mechanism in general, but in order to understand the mechanism, we need to know how the proteins are connected one to the other, and single protein works. As I said before, biofibers. Okay, another information, another tip. This is also another question for the exam. Skeletal muscle. Can you tell me what is a sarcomere and how a sarcomere is constructed? And you just have to show me drawing a sarcomere like this one. Because if you understand how a sarcomere is constructed, for sure you know how the sarcomere can contract. Okay, so as we said before, myofibrils are muscle fiber contractile structure that are composed of different types of proteins. And you can see here all the proteins that we already analyzed before. One muscle can contain 1,000 or more myofibrils that occupy most of the intracellular volume. But this is obvious. It is a function of a skeletal muscle contraction. So inside of a muscle cell, there should be a lot of protein that allows the contraction. So the protein in here are responsible for the contractility of the skeletal muscle. And we need to study one by one these proteins. Okay, so the thin filaments, here, the red salmon color, is constituted by a protein named actin. Okay, you can see here this little globular circle here, that is the actin. Okay, actin is a globular protein. If you see here in the lower part of the slide, multiple G-actin molecules polymerize and form a long chain of filaments called F-actin. So the G-actin, the global actin, G-actin molecules that polymerizes and form a chain of filaments that are called F-actin. You can see here one filament and the other one, like an alpha helices. Okay, like this double strand of F filaments constitute the thin filaments. You can see here, but also here. Okay, this is an enlarged portion of this. And the actin here is made by F-actin. Okay, F-actin, double strand of F-actin. But as you can see clearly, there are also other proteins here. These other proteins are called regulatory proteins. The blue one here, it is called tropomyosin. And the other one here, also in salmon color here, it is called troponin. So there are actin filaments, and on top, regulatory proteins, such as tropomyosin and troponin. We will analyze this protein better later, but I would like to remind you that since they are regulatory proteins, the meaning of regulatory is that they directly regulate the process of contraction in the skeletal muscle. Okay. And now it's just important to remember that troponin and tropomyosin are regulatory proteins, and they directly regulate the process of contraction in skeletal muscle. Finally, there are other accessory proteins, such as the green one here, titin, and the nebulin here, in green, titin and nebulin. They are not regulatory proteins, but structural proteins, and their function is to maintain the proper alignment of the filaments, of the thick and thin filaments, during the contraction, and also give them the elasticity. The titin protein is an elastic protein that gives to the muscle elasticity. So we have regulatory protein, troponin and tropomyosin, structural protein, titin and nebulin. The function of the regulatory protein is to participate in the contraction. The function of the structural protein is to maintain the proper alignment of filaments during the contraction, and also give them elasticity. Thick filament in purple here is made by myosin. The myosin. You can see here the thick filaments, and then here the enlargement of a part of the filaments. The myosin is the real motor protein with the ability to create the movement. Muscles, and these differences determine the muscle speed contraction because they can work in different time. So different isoforms of myosins create different speed contraction. We will understand this point. This is an important point later. Almost 200, 300 myosin molecules join to create the structure, to create the thick filaments, where one myosin molecule is composed of two identical protein chains, each with one large heavy chain plus two smaller light chains. The heavy chain, you can see also here, the heavy chain are the motor domain with the myosin ATPase. The light chain have the regulatory function And this light chain is still under intense study, okay? But for us, for physiology, for us, it's important to understand that the structure of the myosin is composed, one myosin molecule is composed of two identical protein chains, each one with one large heavy chain here, the myosin tail, plus two smaller light chains here. The heavy chain are the motor domain with the myosin ATPase, and the light chain contain the regulatory function. You can see also here the hinge region, it is quite mobile, and the myosin head. The myosin head are important because they can carry to the actin, and this is the real important point of the contraction. I add in this figure that you have in your book, okay, this second part. Why? Because here you can appreciate the myosin heads that contain the light chain, and the actin binding site. This means that the myosin head can connect with the actin in the actin binding site. And second point important is that the myosin head contains here the ATP binding site. We said before that the myosin is a protein that has an ATPase function. What is an ATPase function? Have you already studied the ATPases in biochemistry? An ATPase function means that when you use ATP, you have energy to do something. Okay, so like the ATPase pump. Okay, you use the ATPase energy to do something. Obviously, here we use the ATPase energy to contract the muscle. Okay, but this myosin that has a ATPase function, this is the ATP binding site. Okay, in the actin molecule here, we have the same. So we have, look at the green little point. Okay, here it is all around here in the actin molecules. The green the green point here is the myosin binding site on top of the actin, because they have to act together. So there is an actin binding site on myosin and the myosin binding site on the actin. When the time 10:11, I told you to tell me before. Okay, let's have 10 minutes. Stop. 10 minutes break. Okay. Minutes, huh? Sì, non ha nessun senso. Okay, so the last point was this one. Okay. The myosin head contains the binding site for the actin, and the G-actin molecules on top here contain the binding site for the myosin. So in each part of myosin heads and actin molecules, there is an actin binding site and a myosin binding site. And if you check carefully here, you can see here. Okay, right. Yeah. Exactly. You can see here the actin binding site on top of the myosin head and the myosin binding site on top of the actin molecule. So the actin and myosin filaments are close together in this condition, and the actin binding site is close to the myosin binding site. The only differences that you can see here, okay, is that in the actin binding site for the myosin here, there is the troponin. This protein here is the tropomyosin, sorry, the tropomyosin, and the troponin here. What does it mean? It means that the tropomyosin here in some way block the actin and myosin binding sites. Okay, we will see it again later, but here there is also the tropomyosin that blocks the actin myosin binding site. Each filaments here is arranged so that the myosin heads are in some way clustered together at each end of the filament. In the central region of the filament is a bundle of myosin tails. Imagine these myosin heads and tails all together, not just two, like in this slide, but a lot of myosin heads. Okay, the heads are and the filaments are linked together with the actin molecules. And there is also here a region that we analyzed before, a hinge or zipper that gives the mobility to each head. This head can move in this, like in this way, okay? Because there is here a hinge, you can appreciate it here, that links the myosin tail to the heads. The heavy chain of the myosin head form the motor domain that use energy from the ATP to create the movement. Okay, we will see how the process can promote the contraction. Because the myosin act as an enzyme to hydrolyze the ATP and gain energy, as we said before. And for this reason, it's considered as a myosin ATPase protein. It's obvious that if this protein that hydrolyzes the ATP can hydrolyze the ATP faster or slower, the functional property is to promote the contraction faster or slower, because it depends from the velocity in which the ATP is hydrolyzed. Most of the time, the parallel thick and thin filaments of the myofibrils are connected by these cross bridges that span the space between the filaments. But not all the space is occupied by both filaments, as we said yesterday. In this condition, when cross bridges are formed by the thick filaments and the thin filaments, in this condition, each G-actin molecule has a single myosin binding site, and each myosin head has one actin binding site and one here binding site for the ATP. And the cross bridges are formed when the myosin head of the thick filaments bind to actin in the thick filaments. Every time this myosin head during the contraction can bind this actin and then this actin and then this one and then this one, just moving the actin filaments. If we check this figure here, now we understand why this portion is darker under the microscope, and this one is less darker, because here the two thick and thin filaments overlap. Here there are only thin filaments. So now we know that which are the players in the contraction of the skeletal muscle. Okay, we know the thin filaments, the thick filaments, the regulatory proteins such as tropomyosin and troponin. We know something about the accessory protein, the structural proteins, the titin and nibulin, and we know that the myosin head forms cross bridges with the actin molecules. Just a few words about titin and nibulin. Okay, they are giant accessory proteins. Here you can appreciate better the function of this structural protein. They are structural protein, but they have obviously an important function. Okay, here the titin protein help, okay, the contraction because maintain the structure of the thick filaments that can move on the right, on the right, and on the left between two actin molecules. And then titin provides elasticity and stabilizes, as we said, stabilizes myosin. The nibulin here helps the alignment of the actin. So every structural protein has a particular function. And we already analyzed that the skeletal muscle, we already said a lot of times that the skeletal muscle is organized in sarcomeres. Okay. Now we can define how a sarcomere is organized, because we know already all the proteins, structural and functional and regulatory proteins that take part of the process. Just take a look at this part, Z disc and Z disc here and here. Okay, this Z disc delimitate the sarcomere, and inside of the sarcomere, we will see also other slides later. Inside of the sarcomere, there are thin filaments, thick filaments. In one region, the thick filaments overlaps with the thin filaments. And in the microscope, we see this part like a dark bundle. And this one, in the microscope, appears more clear. Why? Because in the rest condition, obviously, in the rest condition, here are present only the thin filaments. So, let's see, let's see the sarcomere here. This is the three-dimensional array with the, when you can recognize the actin in red here, the Z disc here, where the actin is attached, the myosin molecules here, the thick filament, okay, and a region where these two filaments are overlapped. We already described the proteins that participate in the muscle contraction. Let's see what happened when I watch the skeletal muscle under a light microscope. The arrangement here of thick and thin filaments in a myofibril creates a repeating structure of the alternating light and dark bands. As we already discussed yesterday, light here and dark bands here. One repetition of the pattern forms a sarcomere, the most important unit in the skeletal muscle. The sarcomere is the contractile unit of the myofibril, and we can appreciate here, either in the picture above, the three-dimensional array. The actin, the red one, and the myosin, the violet, form a network of parallel, overlapping thin and thick filaments that are held here in place by the Z-disc, and also we can say also the M line in the middle of the sarcomere. The Z-disc have the filaments here and here. Between two Z-discs is comprise the sarcomere, while the I-band here and here is made of thin filaments only. Here, okay, and this is the three-dimensional structure. The abbreviation I-band, or the abbreviation I, is for isotropic. That means that this region reflects the light uniformly under a polarized microscope. This is the same structure that we have seen at the beginning. When I analyze the structure of skeletal muscle under the microscope, and I use the light, polarized microscope use the light in order to see here, in this region is able to reflect the light uniformly. The A band here, okay, from one end of myosin and the other end of myosin, is made of darker regions where both light and heavy filaments overlap, here and here. For this reason, it's named A, anisotropic. The light is not reflected or less reflected. The H zone here is a clear band in the middle of the A band. Why the light here is clear? Because there are only heavy chain, only the myosin filaments. Finally, here the M line represents the protein to which every filaments, every heavy filaments attach. It's something like the Z disc for the actin. Okay, here in the M line, the proteins, the heavy filaments attach. So the final view of the sarcomere shows that each thin filament is surrounded by 3 thick filaments. Look at this one. And 6 thin filaments encircle each thick filament. This is the this is an important issue that we have to remember, because here and here the process of the contraction takes place. I would like to show you immediately this picture. Okay, wait a second. Okay. Look at this picture here. This is a real picture of a muscle. Okay, this one and this one. And think what the research, the investigators in 100 years ago, when they started to study the muscle, they were supposing that the muscle was like an elastic. Okay, so the contraction is like when you straight the elastic, and the contraction can take place. But the muscle is not like an elastic. You can appreciate here the point. Why it's not like an elastic? Because the investigators at that point, the only important that data that they were able to analyze is that the A band was always dark. So if the muscle behaves like an elastic, everything is going to be more small, and also the A band should be smaller. But in this case, the A band, they start to say, okay, but why the A band remains always at the same dimension? Why the A band never changes? It's not possible. The A band also here always remains at the same level, on the same dimension. Why? The reason is because the thick filament and the thin filaments just move one on top of the other, like in this figure. The thick filament with the myosin head move attached to the thin filament, and the contraction can take place, but the A band remain always at the same level. Actin and myosin do not change length, but they slide one into the other. This is the reason why the I band is smaller after the contraction. Why? Because the myosin and the actin slide past one another. Okay? So here you can see a not contracted muscle, and here a contracted muscle. The A band is the same, has the same dimension, but the I band is smaller after the contraction. Why? Because the actin and the myosin slide one into the other. Okay? And the A band remain more or less the same. Not more or less, the A band remain the same. So, after the description of the structure of the sarcomere, we have to understand how the contraction takes place, because we know the players now, we know the structure of the players, but we still don't know how the contraction takes place. We know that we will use all the filaments and the bands we have just described. And the contraction of muscle fiber is a process that enables us to create force, to move, or to resist a load. For example, I am able to pick up a shopping bag, but I'm also able to carry the shopping bag for some minutes to my home, okay? In one case, the force that I have created moves the shopping bag. In the other case, I just resist to a load of the shopping bag. I can try to move the wall and creating a force, but I cannot move the wall, okay? So a force, a tension is always created. In muscle physiology, this created force is called tension, okay? It's the force created by the muscle, while the load is a weight or load that opposes the contraction of the muscle. So just a little bit of terminology. The muscle tension is a force created by muscle, while the load is a weight or force opposing the contraction. In this context, the contraction is the creation of tension in the muscle. Instead, the relaxation is the release of tension. And now, which are the steps leading to skeletal muscle contraction? There are events at the neuromuscular junction. We already described this point a little bit, but we will analyze deeply now, because we know all the players now in the neuromuscular junction. So the first step is the conversion of an electrical signal, the action potential coming from the motor neuron, along the somatic neuron, in a chemical release of a neurotransmitter. Which is the neurotransmitter released in the neuromuscular junction? You remember which is the neurotransmitter released in the neuromuscular junction? Is the acetylcholine, okay? Acetylcholine is the neurotransmitter released, and the receptor is a cholinergic nicotinic receptor. The second step, so in the events at the neuromuscular junction, we start from an electrical signal with the release of a neurotransmitter, so the release of a chemical signal. The second step is the consequent electrical signal in the muscle fiber, which is fundamental for the muscle contraction. So the neurotransmitter outside in the neuromuscular junction needs to be transduced in an electrical signal in the muscle. The excitation contraction, the EC coupling here, is the process in which muscle action potential initiate the calcium signals. That is the initial activation of the contraction relaxation cycles. We already said that for contraction is important calcium, so this is the initial process in which the action potential initiates the release of calcium. Extremely important for the contraction. Without calcium, the contraction cannot be activated. With a lot of calcium, the contraction is always permitted, even if the stimuli is not present. The calcium needs to return to physiological level after the contraction. And you already know everything about who are the players in order to maintain the calcium concentration to a physiological level. And finally, the contraction relaxation cycle is going to be explained, we will see it soon, by the sliding filament theory of contraction that I introduced you before. The sliding filament theory of contraction is the explanation of why the muscle is not like an elastic, and why the A band remain always at the same level. The contraction relaxation cycle, so the sliding filaments theory of contraction is this one. The A band remain always at the same level because the contraction can take place when the actin filaments and myosin filaments move one into the other through the contraction relaxation cycle, through the sliding filament theory of contraction. We will see in the next lesson on Friday morning, how the process can take place. Okay, because now we are ready to understand, to couple the excitation through the motor neuron and the contraction in the muscle. Question? Okay, so see you next time. Right. I think this is right. Bye bye at home. Have a nice day.